But there are problems with this type of retrospect. So, for example, there's the problem of selection bias. So we know that we're only looking at results from 2 pharmaceutical companies out of many pharmaceutical companies. And also the problem that you're relying on people's their memory of recollecting what's been done in the past, so past experiments and past data. So we've got the problem of recall bias. And there's also the problem with these 2 published studies that the details and data behind these claims are not available. So we could actually question the reliability of these 2 studies. However, there have been prospective studies carried out. So these were academic-led with great attention to detail, and they had adequate statistical power and pre-registration of participants. And across different research domains, we've seen that there's also a failure to replicate studies. So in psychology, we found that only 36% of 97 studies could be replicated. In cancer biology, 40% of 10. In economics, 61% of 18. And in social sciences, 62% of 21. Similarly, we have the problem of selection bias. So these studies are only selecting studies, or could be only selecting studies that they might suspect are not going to replicate. So we have that problem. But we do know that all details and data were available for these studies. So there is more chance of these being reliable results. Due to publications like this and studies like this, there has been an outcry about a reproducibility crisis, where there's an enormous burden with a waste of resources that includes patients and scientists' time, and also delays in coming up with new treatments. So there's been a lot of reporting of this in the press, where we see Nature, for example, have surveyed people to ask whether they think there's a reproducibility crisis, and we can see that yes, 52% of people or researchers surveyed agreed that there was a crisis. And they've also published a comment looking at who's responsible for trying to hold up robust science. So they found or claimed that funders, journals and researchers are all doing their part to try and make science reproducible, but it might be a problem with the institutions themselves, where they're not concerned about this aspect of research. And obviously this is something that we would like to look at more and find solutions for. So just to give you another example of where they've looked into reasons for a lack of reproducibility, and this was done in a study looking at worms. So first of all, many years ago, there was a claim that in one lab, a drug could expand the lifespan of worms. And of course, this was an exciting result that people hoped could be replicated across different laboratories. However, it was found that this didn't reproduce across different labs. And it was only many years later that they set out systematically to test the reason for this. So they looked at over 100,000 worms, and they developed a protocol between labs, and this allowed them to uncover a huge number of subtle disparities in techniques and notations that could underlie this lack of reproducibility. They even found that within a single lab, that a worm could partition into 2 modes of aging, short-lived or long-lived. And this was a within lab variance that they weren't aware of until they started looking at this systematically. This highlighted the importance of replication within a single lab. So as we mentioned earlier, to make sure your results are robust, it's important that this can be found in replication studies within a lab. And they also found it was crucial to understand sources of variability between labs and environments. This table here looks at the different variability and how it's attributable to that source. So we can see that genetic variation is a cause of this variability, and the reproducibility between labs, within labs, and individual variation all contribute to this lack of reproducibility. So looking across these different research areas, we would think that a lack of reproducibility of experimental findings has been observed across a wide variety of settings, so that it can be considered a general phenomenon. Therefore, unless a field can demonstrate that it doesn't have a problem, it is reasonable to expect that it does. So we've used the tools, as I mentioned earlier, of systematic review and meta-analysis to look at this in more detail. So systematic review is a review article which uses a structured process to identify all data relevant to a specific research question, and the criteria for identifying information are determined in advance and are transparent, so that's often in the form of a protocol. Meta-analysis is a statistical technique to combine data from its separate experiments. So within this talk today, I'm going to present some results from some of these research projects. But we can use these tools to assess the quality and range of evidence, identify gaps in a field, quantify relative utility of outcome measures, inform sample size calculations, assess for publication bias, explain discrepancies in preclinical versus clinical trial results, inform clinical trial design, and inform 3R's decisions. So I'll give you some of the results from these. First of all, it's important to think about why systematic review and meta-analysis can be useful for animal data. We find that there's often a huge amount of confusing data. So this timber plot on the right-hand side of the slide here represents the results of 222 experiments, all looking at hypothermia as a treatment for stroke. And the effect size shown on the y-axis is the percentage reduction of infarct volume. So if we use systematic review and meta-analysis to try and make sense of this amount of data. Because if we chose to only look at the right-hand side of the graph, we would be able to say that hypothermia definitely improves outcome in these animal models. However, if we chose to only look at the left-hand side of the graph, these studies here, we would claim that hypothermia in fact makes it worse. So this is why it's important to get an overall estimate of efficacy of this treatment, and also look at the sources of variation. So why this study shows such a good outcome and this study actually shows that it's worse. So this is why that we find meta-analysis and systematic review can help us answer some of these questions. We have observed in experimental stroke that there has been failed translation. So to give you the data behind this, this is a study by O'Collins et al. Published in 2006. She looked at the published studies looking at treatment effects in animal models of stroke, and these were in vitro and in vivo studies. And she looked at those only tested in vivo. There were 603 different studies, and within those, treatments were effective in 374 of those studies. So the animal literature suggested that there was 374 potentially useful treatments. And of these, 97 of these treatments were tested in clinical trials. So you would expect that there would be some concordance between what was observed in animals and what was observed in humans. Unfortunately, there was only one of those drugs that were shown to be effective in clinical trials. So we can see that there's a real mismatch between what we're observing in animal studies and what's observed in the clinic. So this isn't just a problem in stroke research. In fact, this translational failure has been observed in multiple sclerosis. So this is an example of a systematic review published by the CAMARADES group looking at animal models of MS. Animal models of bone cancer pain have also observed this translational failure. In animal models of intracerebral hemorrhage, again, and also in animal models of Alzheimer's disease. This is just a few examples of the different fields that are affected by this problem. So we've had a thing, or CAMARADES as a group are really focused on looking at why this is the case. And we have some ideas as to why this might be the case, and I'll give you some examples of the data that support some of these items. So we think that animal studies can mislead if their conclusions are confounded by bias or threats to validity. So this includes how the experiments were done. So if there's lack of internal validity, for example, poor study design, lack of measures to control bias, this can lead to animal studies misleading with their conclusions. Also low statistical power. If the wrong sample size is used, again, we can be not confident in the result. And there's also the problem of lack of external validity. So this is where the results can't be generalised out with this experiment. So, for example, if the wrong model is used to try and answer a research question for the clinic. We also have a problem of how the data were analysed, and if the wrong statistical test is used, or if there's flexibility in this approach to try and guide results in the way that you would want, then we have a problem there. There's also poor reporting of methodology, and if incomplete data are available, such as publication bias. So we'll talk a little bit about all of these issues in the next few slides. When I talk about internal validity, this is to what extent do the study results reflect the true cause effect of an intervention. And internal validity can be compromised by bias within the study. And this might be shortcomings in study design, conduct, analysis, and reporting. To give a few examples of the types of biases I'm talking about here, one is selection bias. So this is where there's a systematic difference in baseline characteristics of groups at your baseline for a study. So this means, for example, if you have a treatment and control group, there is some kind of bias come into play between which animals were selected for each of these groups. So we can address this by making sure that we randomise animals to groups. There's also the problem of performance bias. So this is where systematic differences between groups and exposure to factors other than the intervention of interest. And this can be addressed by allocation concealment. This means that anyone handling the animals, from those in the animal house to those setting up them in their outcome measure apparatus, are not aware of which group the animals would fall into when we're talking about a treatment versus control group. We also have the issue of detection bias. So during outcome assessment, if people are aware of which group the animals fall into, again, this can influence our results. So we can address this by blinded outcome assessment. And there's also attrition bias. So this is systematic differences between groups in the way dropouts are handled. So we can address this by pre-specifying our exclusion criteria, reporting our dropouts in our publications, and also we can set out from the outset to do intention to treat analysis to address this. Now, these are not new concepts. This has actually been well known for many years that these biases can affect our results. And to give you an example of a study that looked into this study was published by Rosenthal in 1963. He asked 12 graduate psychology students to run an experiment where rats were placed in a T-maze with a dark arm or a light arm. These would be alternating at random with the dark arm reinforced for the animals. And the students were told they had two groups of rats. These were maze bright rats and maze dull rats. The maze bright rats had been bred over many generations to perform well in this type of test, and the maze dull rats were just standard laboratory rats from the animal house. And the students ran this test over 5 days, and in fact they did report that the maze bright rats performed much better in the T-maze. They also reported that the maze bright rats were nicer to handle, less likely to bite, and had softer coats. So the students could definitely see a difference between these two groups of rats. However, it turns out that this experiment wasn't on the rats, but on the students themselves, because in fact, all the animals, all the rats, were from the same cages in the same animal house and were standard laboratory rats. So this result just shows you how your expectation, and hence how you handle the animals in an experiment, can influence the results that you observe in an experiment. As I mentioned, we've often focused on animal models of stroke in the CAMARADES group, and this was published by the group back in 2008, looking at a drug called NXY059 in animal models of stroke. Now, this drug had been taken forward to clinical trials, and there was much excitement about what results it was producing in animal experiments. So it was shown from the animal literature that this was a very exciting drug. There was a lot of investment to put it forward to clinical trial, and unfortunately, at the clinical trial phase, it failed to improve outcome in stroke patients. So CAMARADES chose to look at the reasons for this by looking at the animal publications in more detail. So they did a systematic review where they identified 11 publications testing NXY059 in animal models of stroke. This was results from 29 experiments with data from 408 animals. And when we looked at the meta-analysis, the overall global estimate indicated that NXY059 improved outcome by 44% in these animals. However, when we performed stratified meta-analysis to look at measures to reduce the risk of bias, we can see that studies that failed to randomize reported significantly bigger treatment effects. Similarly, studies that failed to carry out blinded conduct of experiment reported larger treatment effects. And also those that failed to report blinded assessment of outcome reported significantly larger treatment effects. So this trend of those that take measures to reduce the risk of bias reporting small treatment effects could underlie this some of this discrepancy between results in animal studies and clinical trials. And it's not just a problem in stroke. In fact, we've observed across different research domains this type of trend where measures to report the risk of bias are associated, or failure to report these measures are associated with larger treatment effects, so including Alzheimer's disease, multiple sclerosis, and Parkinson's disease. This is all looking at stratifying by blinded assessment of outcome. So we were interested as to whether researchers were taking and reporting these measures across different research domains. So we looked at a non-random sample by looking at the CAMARADES database, and we looked at 2,671 studies, and we looked at the reporting of these measures. So we found that only around 25% of studies reported random allocation to group, only around 30% of studies reported blinded assessment of outcome, very few studies reported sample size calculations, and around 15% of studies reported conflict of interest statements. So it was clear that there was a real issue with lack of reporting of these measures. However, when we presented these results, it was put to us that perhaps studies from universities of higher quality were in fact producing higher quality studies. So when I talk about quality, I'm talking about studies that report these measures. So we actually looked at the data from the 5 best institutions in the UK. So we looked at 1,173 publications using non-human animals published in 2009 or 2010 from these universities. And actually we found that there was even lower reporting of randomization. So only around 15% of studies reported randomization, less than 20% reported blinded assessment of outcome, only 10% reported inclusion-exclusion criteria, and again, very few reported sample size calculations. So we can see from this it's an issue across all types of research that there is low reporting of these measures. The other thing that we think is important is to be using an appropriate sample size. So when we're talking about sample size, this is the number of animals necessary to detect a biologically important effect. The reason for this is too few animals and the results can be imprecise and lack the statistical power to detect meaningful differences between groups. And too many animals actually contribute to research waste. So this is waste of animals, money, and time. So you can see why there's a big impact of carrying out a sample size calculation. However, it's so seldom reported in the literature that we can't really look at whether this is associated with larger or smaller treatment effects. And because sample size is so important, the reason that sample size is so important is because this affects our power. So the probability of finding an effect, if that effect is truly present. And to give you an example of this, this is a image from Mogul and McLeod 2017, where they looked at in vivo studies testing 250 treatments, and they reported the positive predictive value. So this is the probability that a positive result is truly positive. They found that most studies only have a statistical power of 20% and a p-value of 0.05. This means that many more false findings of where a PPV of only 50% are reported. So this means that 20 preclinical studies showed promise and were published, but 10 of those were actually false positives. So there's only 10 truly positive studies. So this is where we're using a sample size of only 10 mice per studies, and it just shows the scale of the problem where we can't really trust these findings if people are not appropriately powering their experiments. Just another thing to highlight when we're talking about sample size calculations is that some people actually are very open about reporting how they decide how many animals are used in their studies. So in this example, we see for optogenetic activation experiments, cell type specific ablation experiments, and in vivo recordings, we continuously increased the number of animals until statistical significance was reached to support our conclusions. So it sounds like these researchers would just have kept adding animals into their study until they got the result they were looking for. So as you can probably tell, this is not an appropriate way to decide on your sample size. You have to decide in advance the number of animals to use depending on your effect size you're looking for and the power you require. So some take-home messages from this first section is good experimental design. There's key components to this. We would expect a hypothesis to be stated, for the experiment to have appropriate controls, for them to use an appropriate sample size and report how they reached that sample size, to take measures and report them to reduce the risk of bias, and to perform suitable statistical analysis. This all together can increase the validity of a scientific result, hence increase the robustness of the science and how much we can take home from it. As I mentioned, there can also be issues with the external validity of a study, and one example of this is the choice of model. So we know in stroke experiments, they often use young, male, healthy rats to test an intervention in animal studies. However, when we're looking at clinical trials, it's often patients who are not healthy, who have comorbidities. So for example, in the NXY059 trial that we talked about, 7% of animal studies used hypertensive animals. So those animals with a comorbidity of hypertension. However, in the patient, so the failed clinical trial that I talked about, we found that there were 77% of the patients were hypertensive. So again, we can see there's a mismatch between the type of so the choice of models, the animals that are used, and those patients that the drug will be tested on. So this again gives reason for the discrepancy between animal and clinical trial results. Similarly, in a study looking at TPA as a treatment for stroke, 9% of animals had hypertensive animals, and actually in those 9%, the drug actually made outcome worse in those animals compared to normotensive animals, so those with normal blood pressure. So this led to, because of this systematic review and identifying this issue, it actually led to when they were designing the clinical trials to exclude patients with hypertension because we knew that this was the case. So this just shows how animal studies, if done correctly and reported correctly, can actually inform clinical trial design. One thing to flag up at this stage is standardization of animal experiments. So from what we've described in terms of the lack of reproducibility, it has been suggested by some that it's good to try and keep all variables the same if we can between different laboratories, and this would ensure that things replicate between labs. However, this study by Werbel's group has actually provided evidence that excessive standardization is a source of poor reproducibility because it ignores biologically meaningful variation between animals, between treatments, between the variables that you're looking at in a study. So this means that they used a simulation based on 440 preclinical studies and compared the reproducibility of results between single lab and multi-lab studies. They found that single lab studies produced large variation between study results, whereas multi-lab studies produced more consistent results, thereby increasing reproducibility without a need for larger sample sizes. So this is one study that actually supports the idea that we should be doing things across labs when we're testing interventions, and trying to introduce or at least keep a little bit of biological meaningful variation so that we can be more confident that this result can be reproduced either in a different lab or a different species, or ultimately from animals to patients. Another threat to reproducibility that we mentioned was publication or reporting biases. So when I talk about publication bias, this is where neutral and negative studies often take longer to be published, or they more likely remain completely unpublished. It means that they're less likely picked up by systematic review, and also leads to the overstatement of efficacy in meta-analysis. This is a real problem with how we do experiments at the moment, and anyone that's done an experiment where they didn't get the result they would expect will maybe have come across this, where you're less likely to put the effort into publishing this and putting it in the public domain. There's also the problem of selective outcome reporting. So this is where you may report if you're testing multiple behaviours, for example, in an animal model of pain, and you find that a drug has a positive impact, so relieves pain in one type of behavioural outcome but not in another, it might be that you're more likely to only report the one that had the positive outcome. So again, this is a way of a reporting bias where we're not sharing all our results in the public domain. And finally, there's the problem of selective analysis reporting. So this is where it might be that we run 2 different statistical tests and only report the one which supports our hypothesis. So again, we're introducing these biases with what goes into the public domain and not sharing the full picture. So we'll look at some solutions to this in some of the summary slides. Just to give you some results from the animal literature looking at publication bias. This again was done in animal stroke studies, and the study by Senna et al. Showed that publication bias leads to a major overstatement in efficacy. So this here is a funnel plot where each black dot represents an individual study, and we've got precision on the y-axis and effect size on the x-axis. And we would expect a funnel plot to have an equal number of positive and negative studies. And when we use trim and fill analysis, we can see that there's actually a lack of imprecise negative studies. So these are the ones that we think remain unpublished according to this type of analysis. And in fact, it's claimed that 16% of experiments remain unpublished, and this leads to an overstatement of efficacy of 31%. So you can see how this, again, publication bias is contributing to overstatement of efficacy in animal studies. So for the next section of my talk, we're just going to talk briefly about some potential solutions. So one of those potential solutions includes reporting guidelines, so how reporting guidelines can make us more open about exactly how we performed an experiment, and hopefully makes things more reproducible because people know exactly what's been done in an experiment. And there's also ways to increase the transparency of research. So this includes pre-registration of studies, so telling people exactly what you plan to do in a study, what analysis you plan to run, what outcomes you plan to measure, so that we know when the final publication comes out that this has been planned from the start rather than being changed halfway through. And we also have the approach of open data, so sharing the data behind statistical analysis so that people can perhaps rerun analysis or look at exactly the causes of variation between studies. To give some examples of reporting guidelines that are out there, we can include the CAMARADES study quality checklist, which was actually designed for systematic review looking at animal models of stroke. So this study quality checklist actually informed some guidelines for the stroke research community, which I'll talk about. But just to go through these in order, we see that this checklist looks at whether it was published in a peer-reviewed journal, whether there was randomization, allocation concealment, blinded assessment of outcome, if animals with comorbidities were included, if researchers performed and reported a sample size calculation, if there was a statement of compliance with regulatory requirements, and if there was a statement regarding possible conflicts of interest. So this actually fed into these good laboratory practice guidelines, and in fact there's seen a huge increase in the reporting of these measures in the stroke research literature. So one example of this is a systematic review looking at IL-1RA as a treatment for stroke, and they found that from the original study, the original systematic review published in 2009, there was a large increase in the reporting of all these measures in the updated version of that post good laboratory practice publication. So there has been, these have led to an increase in the reporting of these measures. There's also the ARRIVE guidelines, so anyone that's carried out any in vivo animal work might have looked at these as a way of, these are a guide to how you report your research. There are a lot of questions, so there's 20 different questions with different subsections, but these have been used to try and guide how we report our animal research. And Nature Publishing Group and other publishers also have published their own guidelines to try and tell authors what they would like in their reporting standards. Now, the question is whether reporting measures alone, reporting guidelines alone, are enough to actually increase the reporting of these measures to reduce the risk of bias. So the CAMARADES group have performed 2 studies to try and answer that question. One looked at the reporting of the ARRIVE guidelines and looked at all manuscripts submitted to PLOS ONE, and this was a randomised study, so any study reported submitted to PLOS ONE was randomised to either treatment group, so these were asked to fill out an ARRIVE checklist, or control group, where they were not asked to do this. And the primary outcome was to look at the compliance and control and intervention groups. So compliance meaning how many of these ARRIVE guidelines did they actually end up reporting when they were asked to. And we found within this study that of the 101 different items that they were asked to report, there was no significant improvement in 100% compliance, which you might expect because covering 101 different items in your reporting is quite a big task to take on. So we also looked at a different study. So this was looking at the Nature Publishing Group guidelines, and the objective was to determine whether a change in editorial policy is associated with improved reporting of measures which might reduce the risk of bias. So the primary outcome in this study was looking at the change in proportion of publications describing in vivo research reporting just 4 measures, so looking at randomization, blinding, sample size calculations and exclusions. So this more specific list, we found that there was an increase in reporting of these measures not seen at other journals. So Nature did in fact increase the reporting of these measures. And I think these 2 studies just show how a more specific targeted approach, so where we're only looking at set items that we think is important, is more useful than something asking for too much information. So I think what this maybe has fed into some kind of policy changes in the Nature group definitely is to try and focus on some of these important measures to reduce the risk of bias. There's other improvement strategies available. So there's a thing called, or an online software called the NC3R's Experimental Design Assistant. And this is designed for researchers starting in vivo research to go on and design their experiment from scratch. It also makes it more transparent on where a sample size came from, because you can use the Experimental Design Assistant to calculate that for you, and also makes people think about introducing blinding, randomization, and other measures to reduce the risk of bias at the design stage. It can also, another approach is to study to publish a protocol, or at least share a protocol. So make this publicly available, which sets out in advance what your intended experiment is and how you, in your experimental design that will be behind that. There's also new publication policies, such as those I mentioned from Nature Publishing Group, but also at PLOS, and the use of things like bioarchive and registered reports. These are all ways of making sure that we publish our data and put it out in the public domain, even if it's not what we expected from a experiment. So registered reports and bioarchive are ways of publishing online without the need for going through the peer review process. So you can put it up there without the need to go through the peer review process and the time that takes. We also would be keen to emphasise rigour in grant award and promotion, so not just look at the high impact factor journals, but also look at these ways that you're sharing your data and perhaps putting your data behind your publication, so this kind of open data policy. So make sure that this is awarded in grant awards and also promotion within universities. And we also try and advocate that not just publishing in journals of the highest impact, but again, making sure that just your research is out there. We're interested in continuing professional development opportunities for scientists. So even if a scientist has been around for a long time, making sure that there's courses available that can help improve experimental design, and highlighting these different tools available to help in research design. And we'd also encourage anyone to systematically review available data from a field before they design an experiment, so you know what's been done before and you can see where the gaps in the field are, so that this can help make sure that we're designing experiments in the best way. Now, for the practical exercise that I mentioned, we're going to look at some critical appraisal of published studies. So this is, I'm just going to go through an example publication first, and then we'll give you some time to look at the papers that I've mentioned. So this study is actually a study in neuropathic pain animals, but I'm just going to show you how we would, at the CAMARADES group, try and critically appraise this publication for systematic review. So one thing that we would look at is whether the study reports randomization to group. And in this paper, we can see, I've taken out an extract from the text, and we can see that the researchers report that rats were allowed to climate to the testing chamber for 30 minutes and then randomly allocated into experimental and control groups. So this is a good example of where they have used randomization. Again, in the same paper, we can see that the investigators were blinded to the drug administered, so again, an important measure to reduce the risk of bias. The experimenters also told us why animals were excluded from the study. So rats showing a post-surgical neurological deficit were excluded from the study. And they also included a statement of compliance with regulatory requirements. So that's just some examples of how we could check the internal validity of a study by looking at the reporting of measures to reduce the risk of bias. For the practical exercise, we have two papers. So the first paper is by Bravo et al., published in 2011, and the second paper is by Prodillo et al., published in 2017. And what I would like for this part of the session is for you to sit in groups of 4, where at least one of you has access to either a phone or a laptop with internet access, so Wi-Fi access, so that we can, when it comes to feeding back on the critical appraisal, you can fill out just a quick quiz online where I'll send you the link at that time. So the things that I would like you to go through for these papers, I'm hoping that these have been made available as paper copies for you. The critical appraisal details that we would like for you to have a look at include looking at this quality checklist. So do the studies report randomization, allocation concealment, blinded assessment of outcome, sample size calculations, exclusion criteria, and whether they report whether animals were excluded from analysis. But also think about, are there any other measures taken to reduce bias in the study? Are there any other risks of bias in the study? How reliable do you think the results of the study are? and how generalizable do you think the results of the study are? So can I ask you all to go into your groups of 4, make sure you've got a copy of each of those papers, and for the next 40 minutes, we'll just give you time to go through these papers, and I'll be online, so feel free to ask any questions to the microphone if they come up while you're looking at the studies in detail. Thank you.